Hi guys. We'll talk about bar body formation. So in bar body formation, what you need to know that this was observed. The bars are observed as darkly stained body in interface nerve cells, and it was found in interface nerve cells which was absent in similar cells of males. Okay, it was present in female cats but absent in case of males, and this body was described as sex chromatin body or bar body. Okay, and the bar body, it is one of the two inactive X chromosomes in mammalian female cells. One of the X chromosome is inactive, and this inactive ones remains condensed during the interface, and it is silent in transcription, whereas other X chromosome is decondensed and transcriptionally active. One is condensed and inactive, another is condensed and active, decondensed and active. So the number of bar bodies present in an organism or a female, it depends on the number of X in a cell. It is n minus 1 rule is followed. Now n is number of X chromosome. Okay, so for example, Turner syndrome, which have only one X, there is no bar body, but where uh, in Klinefelter it has two X and one Y chromosome, which is in males, Klinefelter males. Here there is one bar body. Okay, next is mammalian X chromosome. Next is mammalian X chromosome inactivation. It is initiated from X inactivation center or XIC. So this is where it is initiated. It is initiated in the region XIC, which is usually found in the near the centromere. Okay, in the XIC region near the centromere, where the inactivation is initiated, the X inactivation center, it contains 12 genes, 7 of which code for proteins, 5 of which are untranslated RNA. Okay, so 7 which will code for protein. So X inactivation center or XIC region contains 12 genes, 7 codes for proteins, and the 5 for untranslated RNA. Okay, and among the 5, only 2 are known to play an active role in X inactivation process. Okay, only 2 play an active role in X inactivation process. Which two are there? Which two untranslated RNA plays role in X inactivation? It is XIST and TSIX. Okay, and this center is also important in chromosome counting, which ensures that random inactivation only takes place when two chromosomes are present. So XIST, TSIX, they both are also involved in chromosome counting, which will count the number of X chromosome, and accordingly bar body formation will take place. So the role of this XIST, TSIX, they appear to be antagonistic. When TSI expression is lost, try to understand, when TSIX expression is lost, okay, lost on the future inactive X chromosome, it results in the increase in level of XIST around XIC. So whenever this TSIX level is lost or reduced, okay, it's lost, when this level is lost, then what will happen? It will result in an increase in level of XIST level will be increased, okay, around XIC, which is the inactivation center. So the future active X, okay, TXIX levels are maintained. Okay, so TXIX level should be maintained. Does the level of XIST remain low? Okay, so XIST remain low when there is when the X chromosome is active. Okay, and when it is inactive, TSIX is when it is inactive, XIST is more. So in case of inactive ones, in case of inactive chromosome XIST, what will happen? It will be In inactive chromosome, XIST level will be high. In case of active chromosome, XIST level will be low. So remember, XIST is what it is doing, it is lowering or it is inactivating the X chromosome. Clear? So next is Lyon's hypothesis. The 2X, okay, one line remains. So does the level of XIST remain low? This shift between XIST, TSIX, it allows to begin coating the future inactive chromosomes spreading out from XIC. So when XIST is high, it will kind of coat the X chromosome, making it inactive. Next is Lyon's hypothesis. So the 2X chromosome in the female cell drive the male gamete. Okay, and from the female gamete, okay, 2X chromosome in the female cell derive from the male gamete and from the female gamete. Okay, so XM male, XP from parent, like kind of paternal, and they are functional in every female mammalian embryo. So once cytodifferentiation starts in the embryonic tissue, the X chromosome inactivation occurs. Okay, when the cytodifferentiation and kind of cell differentiation starts, there is a random inactivation. We doesn't know that which X chromosome, be it paternal or maternal, which one will be silenced. We don't know. And this random inactivation of XP, which is paternal, like XM, which is maternal, in the somatic cell during embryonic development is known as Lyon's hypothesis. Okay, and this occurs in all eutherian mammals, making females functional mosaics of heterozygous X-linked genes. So since it is happening in all mammals, all eutherian placental mammals, which is making females functional mosaic, okay, of heterozygous X-linked genes. In marsupials, the paternally derived X chromosome is always inactivated in the female animal. Okay, so it depends upon different organism. If it is eutherian or placental mammals, there is it is a mosaic formation. Somewhere X paternal is inactivated, somewhere X maternal is inactivated. And in case of marsupials, always the male X chromosome is inactivated. So X chromosome inactivation, see the picture, the maternally and paternally derived X chromosome, they are represented as XM and XP. So in case of XM and XP, you can see that the gamete will be formed, okay, further gametes will be formed, and X chromosome inactivation will be there. Somewhere you can see here, here XM is active and XP is inactive. Here XP is active but XM is inactive. Okay, in the another case, same thing can happen. Here XP is active, XM is inactive, and the same way. So bar body is formed from the inactive chromosome. Next is dosage compensation. So dosage compensation is what? So two chromosomes are there in normal human females and only one X chromosome in normal human males. Okay, and which is unique compared with equal number of autosomes present in the cells. Both have equal autosomes. Okay, but X chromosome, one in male and two in female. Okay, in case of human. And since females have two copies on the X chromosome and males have only one, okay, then there is a potential for females to produce twice as much of each gene, product for all X-linked genes. If both the X chromosome will be active, then females can produce twice as much gene, okay, as males. So dosage compensation is important, which is the activity by which X-linked gene is made equal in the two sexes. Okay, X-linked genes which is expressed should be equal in the two sexes, XX and XY. In placental mammals, marsupials and some monotremes, compensation is achieved by inactivation of one chromosome in somatic cells of females. And in case of Drosophila, where X chromosome is not inactivated, both X chromosomes are transcribed in females. So compensation, how it occurs in Drosophila, it occurs by making the single X chromosome in male hyperactive. So male is X0, it makes single X chromosome hyperactive in case of Drosophila, so that they can be like compensated. And females are not so hyperactive, but both XX are expressed. Okay, as a result, the level of transcription of the genes carried by X chromosome is same for both sexes. So that was about Barr body, Lyon's hypothesis, and dosage compensation. Very important topic to understand about. Next is packaging of DNA into chromosome. Okay.